next to hear from Dr. Singh on some new plasma biomarkers.
Thank you. So I thank the organizers for inviting me. This is the first time I am attending this meeting. Especially thank Ali Fatmi. So this work also is in collaboration with Candy Krieger. So I'm a basic scientist. So most of my studies have been on the disease mechanism using the mouse model mostly, and this is kind of the first patient tissue or plasma-based study that we are carrying out. So I'm going to skip a couple of first slides because most of us know what we are talking about here in terms of the disease pathology. So this, I'll be focusing mainly on the biomarkers. So before that, I'll show some of the data from the post-mortem brain tissue that we obtained from the NIH brain bank of the ALD patients to kind of generate a proof of concept for the study of these biomarkers. So the objective of this study was to identify a panel of plasma biomarkers that can predict. In this case, the data which I'm presenting is AMN versus the cerebral AMN, and then the study related to cerebral ALD are ongoing in our lab. So there are two molecules or two compounds, you can say, I'll talk about, which are microRNA and the metabolites. So I'll just kind of introduce first what these are. So what is a microRNA? It's a small non-coding RNA, is just like we have the normal RNAs which are converted into the proteins. But these microRNAs are, as the name suggests, smaller than the regular RNAs that code for proteins. And their activity especially is to repress the gene functions. So their role has been quite well documented in the regulation of gene expression during development. So but now their focus has also come up in the progression or regulation of the diseases. So these microRNAs, they regulate about 30% of the mammalian gene products. And one microRNA can target multiple mRNAs. So there are lesser of microRNAs and more of mRNAs in a genome. And they have been described in every kind of species. And their sequence is very conserved. So that kind of basically helps also with determining their functions. So they are called the dark matters because they are present in the non-coding region of the RNA, and there are different microRNAs that have been classified. The microRNAs which decrease the mRNA stability. There are certain different type of RNAs, pVRNAs, that regulate the epigenetics or the epigenetic regulation of the gene function. And then there are different promoter-related microRNAs that regulate the gene silencing and chromatin or the chromosome modulation in the species. So why you want to study microRNA? Basically, from your DNA, about 85% of the DNA is transcribed into a RNA, but only 1 to 2% of RNA is translated into proteins. So rest of the RNA, which is present in the intron sequences, which are the non-coding sequences, those contain mostly the microRNAs and long non-coding RNAs. And so now previously considered a dark matter, the way it was named, because no function was assigned to it. Now we know that these microRNAs and long non-coding RNAs play a very important role in gene expression, both during the development as well as during the disease. So microRNAs are not new. They were described almost 2 decades ago. And in fact, the first description was from this city, from Harvard Medical School. And now they are implicated in terms of diagnosis, early diagnosis, staging of the disease, as well as prognosis, follow-up, and also to monitor the therapy response. But if you identify a panel of biomarker or panel of microRNAs that change with the disease progression. So, and I just put a couple of papers here, but there are hundreds and hundreds of manuscripts on the role of these microRNAs, including in the neurodegenerative diseases. So especially here I put on the neuroinflammatory diseases like the multiple sclerosis. They have shown in ALS, they have shown Huntington. So in XALD patient lymphocytes, you see expression of the inflammatory genes, especially NOS2, which was also mentioned today morning, nitric oxide synthase. So we also saw that these microRNA in those lymphocytes can regulate the expression of those NOS2 genes. And then we have also shown these in the MS mouse model to play a role in to be associated with the disease progression. The second thing which I mentioned was the metabolomics. So basically you have, this is the standard pathway of gene translation in any cell. You have DNA to RNA, proteins, and then these proteins under various effects, what we call as the environmental factors, these metabolites respond to those environmental factors and are changed based on the environment or the disease of a patient. And the miRNAs come in here. So these miRNAs, they can block the translation or inhibit the translation from RNA to proteins. So that is how these miRNA and metabolomics are linked, or the metabolites are linked. So we wanted to see what happens in this disease in terms of miRNA and metabolomics. So metabolomics also has been shown in a number of diseases, including the neuroinflammatory diseases, both as biomarkers as well as the mechanism of the disease progression or remission. So in generating the proof of concept, as I mentioned, we use the postmortem brain tissue from cerebral ALD. So what we did was, within the cerebral ALD, you have areas of demyelination and areas which are completely normal looking despite the accumulation of very long chain fatty acids. So we took those brain white matter areas which were normal looking, as well as the plaque shadow, or the areas, inflammatory areas surrounding the plaque, and the age-matched control brains, postmortem brains, and then these were subjected to metabolomics and mRNA sequencing studies. And whatever the changes came, they were bioinformatically analyzed for their significance. So I'll skip the data analysis. So this is basically the whole kind of a genome sequencing and metabolomic study what gave us. So first thing that shows you is that these samples which we took are clustering independently and not overlapping between the control and the normal looking area and the plaque shadow area. So indicating that they have clear, distinct expression of microRNAs or metabolites in them. And heat map shows you what kind of expression differences there are between the samples. So you can see the controls versus the normal looking area from the CALD brain, and then the plaque shadow, they have a very distinct expression of the microRNAs. And running the bioinformatics and biostats programs just tells you what kind of microRNAs are distinctly changing with the disease progression in these samples. And the same was done with the metabolomic analysis. So narrowing it down, we just focused, there were a lot of microRNAs and metabolites that changed, but we only focused on the microRNAs and metabolites that showed a pattern with the disease severity. So either they increased with the disease severity or they decreased with the disease severity. So the disease severity here is control to normal looking area to the plaque shadow of the cerebral lesion. So, there are, I'm not sure if these are readable. So there were 19 microRNAs that we found that changed with the disease severity. Out of these, 17 were increased as the disease severity increased, and there were 2 that were decreased. So significantly in these, those who follow the microRNA literature, you can see that microRNA 155 and 146, and there are a couple of 146 and microRNA 27 which are related to inflammation. They have been documented by multiple studies, both in mouse model and humans, to be related to inflammation. They were increased with the cerebral lesion going from control to normal looking area to the plaque shadow area. And there were 2 microRNAs that were significantly decreased with the disease severity. And of these, I think the most interesting for us is the microRNA 338, because multiple studies have shown that microRNA 338 regulates the oligodendrocyte development. So oligodendrocytes are the myelin forming cells in the brain. So the more oligodendrogenesis, the more you have myelin. So because these microRNAs were decreased, and this microRNA 338 regulates the oligodendrogenesis directly. So more the microRNA 338, more will be the oligodendrogenesis by the oligodendrocytes. So we found that this microRNA 338 was decreased when in the areas of inflammation, but not in the normal looking area from the cerebral ALD brains. When we looked at the metabolites, we found about 7 metabolites that were increased with disease severity and 6 that were decreased. And again here, there were not many surprises, because metabolites like butyl carnitine and hexonyl carnitine, they are the products of incomplete beta oxidation, which obviously occurs in ALD. And then palmitic and stearic acid have, especially stearic acid has earlier been shown, although quite long ago, to be involved in the cerebral ALD, in the ALD pathology. In terms of decrease, we saw the myelin-related proteins or metabolites like diphosphocholine, 16-PC, or glycophosphocholine, which are the components of the myelin in the brain. They were decreased as the disease severity progressed in these brains. So seeing that these microRNA and metabolites do respond to the disease severity in this, so what we proposed a grant to the NIH to look at the patient plasma samples to identify biomarkers that can be associated with the disease severity in the patient samples. So we collaborated with Candy Krieger, from where we got the plasma samples. So the first group of study was between control AMN and cerebral AMN patient plasma samples, and the samples were sent for metabolomic and miRNA studies by RNA sequencing. And again, looking at the biostat and bioinformatic analysis, the plasma samples separated into clear distinct groups. That means that the expression of metabolites and microRNAs could separate these plasma samples into clear groups based on their disease severity. So, and the pathway analysis in the metabolites, so we compared the control with the AMN and the AMN with the severe or the cerebral AMN to see which kind of metabolite pathways are changed in these metabolites. So the pathways most changed between control and mild were nitrogen metabolism and glycine, serine, and alanine aspartate pathways, they were common between the mild and the severe comparison. But as we'll see in the later slide, in terms of the metabolite expression, there was a clear distinction, separation between the mild and the severe plasma samples. So the glycine and sarcosine metabolites uh, were increased in the with the severity of the disease, and the alpha ketobutyrate was decreased to because these analyses when you go on for different batches of plasma samples can vary based on batch to batch variation. So what we devised was to take a ratio of the metabolites within the same pathway. So we expect that the ratio or the degree of change of the metabolite will likely be more stable than the expression or change in the individual metabolites between different disease phenotypes and samples. So the ratios are consistently show that the glycine sarcosine ratio decreases as the disease severity increases, and whereas the threonine alpha ketobutyrate increases. So, so this and then a second set of the pathway which was changed was the aspartate and the glutamate pathway. And again, it shows there is a clear distinction as the disease severity progresses between the expression of these metabolites. The second set of studies in this was to look at the RNA-seq or the miRNA expression. So when we looked at the control versus the AMN, there were total 36 microRNA in the plasma that were altered. And when we compared the severe and mild, there were 30 altered. But then again, we looked at those microRNAs which changed with the disease severity, either increased or decreased. And it was good to see that we could narrow it down to 3 with the disease severity, and those 3 miRNAs were 134, 186, and 409. And looking at the literature, this is quite interesting because MIR, all these microRNAs have been functionally tested in at least some of the models to show what kind of genes or metabolites they target. And MIR 134 5P has been shown to regulate the neuropathic pain, and MIR 186 and 409 are regulated with inflammation. So this, especially MIR 409 3P, is of considerable interest going forward looking at the molecular analysis, because this is expressed in the astrocytes, which are the most abundant cells in the brain and also create the inflammation in addition to the microglia. So this 409 3P has been shown to regulate the IL-17 and couple of IL-1 beta, IL-1R receptor cytokines. So these cytokines are known to play a very significant role in neuroinflammatory diseases. And at the same time, this 186 5P was also shown to regulate the neuroinflammatory mechanisms in the brain, in the CNS cells, or in the brain in a couple of models. So overall, it shows that although it's in some of the expressions of the metabolites and miRNA, which are more closer to the control pattern, shows that there is an intermediate expression of microRNA and metabolites with the disease severity, and it changes as the disease severity increases or decreases. So the metabolites may act as a reliable plasma biomarkers to monitor the disease progression. Of course, this involves longitudinal as well as the prospective studies to see whether these microRNA change as with the time. And we believe that the multiparametric metric biomarkers are more convincing and more sensitive than single biomarkers. Therefore, combining microRNA and metabolites makes more sense. And microRNA and metabolite-based diagnostics probably can be used to follow up the disease progression in AMN and cerebral ALD. So ongoing studies are that we are looking at we are going to look at the mRNA and metabolites in cerebral samples, and probably based on these results, we plan to continue our collaboration with Candy Krieger to look at prospective studies, and at the same time look at the targeted mRNA and metabolite molecular studies in cellular models to identify their target genes and proteins to study the mechanism of the disease progression in AMN and cerebral ALD. So I thank the Kennedy Krieger Institute for the valuable samples they provided, and also the funding from NIH for this study. Thank you.
Thank you, Jaspreet. Any questions? You didn't raise your arm, so I'm going to go there first.
Do you have any sense as to whether the metabolite changes are causative, or are they a result of the disease progression?
So at this point, because these are one-time point samples, and both in the brain, it's from the postmortem brain, so it's difficult to say whether these expressions are causative or the result of the disease. So I think more will be that's more answerable when we do the longitudinal studies to see. So here the idea is to first was to see the feasibility, which we definitely see that these microRNAs and metabolites are feasible to be used as the biomarkers, and is more of a proof of concept. And then because there are about 1100 or I think 1700 microRNAs in human genome which have been identified till now. So you definitely need to narrow down to a certain set of panel which you can then test either prospectively or longitudinally to see whether any particular set of microRNAs from that are causative or predictive of the disease phenotype.
Yeah, great novelty here. I think you're a log too low in your sample numbers.
Yes,
we are. You really cannot say anything at this point.
Yes.
So I think that's a difficulty here. You really want to expand it, and then I think, as you outlined, the prospective and longitudinal studies are the key. But it's you know this field better than I do, but you're going to find a lot of things, and
yes.
Who knows what it's related to.
Yes.
So yeah.
There's a question here and that one in the back.
Thank you. It's actually very refreshing to see that somebody is going after multi-parametric evaluation of the disease, because oftentimes people are trying to identify that single marker, and anybody who studied samples and did metabolic profiling knows that there are no cases when one marker is changing and nothing else. I have two questions. One is the examples that you have shown are typically metabolites of high abundance. Was sensitivity of LCMS method a factor in this experiment, or you had variety?
I think sensitivity was not a factor, because there are two broad methods for metabolite analysis: the LCMS, GCMS, or the NMR. So LCMS, GCMS is the most which can identify the most number in terms of metabolites, and NMR is restrictive in numbers, but it's more specific. So in terms of metabolites, I don't think LCMS was restrictive in terms of sensitivity. So, and then these metabolites which we identified are quite relatable to the disease mechanism, I would say, in terms of like glutamate changes or the changes in glycine or alpha keto glutarate, which are more related to the mitochondrial function, which is seen in this disease, the mitochondrial dysfunction. So I don't think the method was any restrictive in terms of sensitivity.
And my second question is a follow-up on your last comment. Have you ever tried to do pathway enrichment analysis based on your metabolomics data to see which pathways are more impacted than others?
Yes, so that pathway enrichment is what I showed here. So this one. So based on here, so when we see that the alanine, aspartate, and glycine metabolism is changing between these two, so then we looked at the individual metabolite changes within these pathways. We looked at which metabolites within these pathways are changing up or down with the disease severity.
Thank you.
Well, I just had a question about like, well, you said that you tested both like healthy and those with ALD, and you've seen like how the microRNA, how their expression has changed from those that are healthy versus those that have ALD or CALD. Have you seen that? And you seen that expression?
And we have not done in CLD yet, so and that's our next aim. So that's an ongoing study. So right now we have finished with the AMN versus the cerebral AMN.
All right, thank you.
Thanks.
Thank you. Thanks a lot. Thank you.